It gives me a great deal of pleasure today to introduce Laura Kiessling. Laura is a Wisconsinite, and she started her education at the University of Wisconsin for 1 year and then transferred wisely to MIT. At MIT, she excelled in 2 areas. She was interested in synthetic chemistry even then with Bill Rausch, but she also spent as much time, as I understand it, on the river, the Charles River. Hopefully she didn't end up in the Charles River, but she was a rower. After her time at MIT, she moved to Yale University, where she worked for Stuart Schreiber, again focused on synthetic chemistry and doing total synthesis of anti-tumor antibiotic molecules. And subsequent to that, she moved west to work with Peter Durbin at Caltech, where she started really thinking about molecular recognition with DNA and forming triplex DNA. And then she decided she wanted to have her own academic career, lucky for us all. And she got a great job at the University of Wisconsin in Madison. I was there, and I can tell you it's a great place to do science. And she started her own independent career, and I think the one thing that was a hallmark of her independent career was using her synthetic background to solve problems at the chemistry-biology interface. And she's worked sort of in two areas using this approach, and she's done very creative science in both of these areas. The first one, she uses the Grubbs method of making polymers, where then she can synthetically dangle a bunch of different kinds of ligands onto these polymers. And a major unsolved problem in biology is that most cells, whether you're a bacteria or whether you have human cells, have receptors that interact with different kinds of ligands. And these ligands cluster, so they have some kind of external signal, and she can control those signals, the spacing, the size, the shape, making these things cluster, and then they have some kind of consequences inside the cell. And so she's built these really creative tools to allow her to study these signals inside the cells. And 2 important areas she's focused on are immune synapses and also chemotaxis in bacteria. Great thing to do at Wisconsin, given Julius Adler was present there. And then the second area, it's probably not the second area, but since it's an area interested in what I read about, is she's become an enzymologist. And I think she's going to talk about that today, where she's focused on designing inhibitors to kill Mycobacteria tuberculosis, a major health problem. And she's uncovered, looking at the biosynthetic pathway of how this complex cell wall around mycobacteria is formed, she's uncovered a novel flavoprotein mutase and novel chemistry that people hadn't anticipated. And today I think she's going to be talking to us about how these weird polymerization reactions actually work and what's the mechanism of initiation, elongation, how do you control chain length. Okay, so that's my introduction, but now I need to tell you all the honors that she's won. She's won everything you can win, just about. But her most famous one, of course, is she's won the Genius Award, the MacArthur Award. She's won many ACS awards. She's a member of the National Academy and the American Academy of Arts and Science. And I think finally I'd like to close by saying she's a terrific role model. Probably her greatest science is her daughter, Kira. And if you ask where the name Kira came from, K-I-E and R-A. Her husband's name is Rains, and she's Kiessling, and K-I-E-R-A, and it means apparently shining light. So congratulations, her daughter is fantastic.
Oh, this isn't my first slide, actually. So let me just say, after listening to that introduction and hearing Joanne say that I'm an enzymologist, that means more than any other award, because that is a huge compliment. So thanks for that really kind introduction. And Joanne explained I'm going to try to explain my science as clearly as Joanne did. So I thought I would start with this picture just to acknowledge the contributions of the Dreyfus Foundation to all of our teacher-scholar lives. And I was listening to the talks today, and the talks, of course, are really diverse. And we have two to go, but those I can anticipate are going to have something to do with the others, which is an interest in catalysis. So catalysis has been a theme from Milan's talk, and Colin showed us some beautiful ways one can use catalysts to design new materials. And I'm going to talk about some enzymes that catalyze reactions, but the Dreyfus program has really catalyzed, I think, our careers, because that recognition at the stage where you're actually being evaluated for not what you might do, but what you've actually done, I think makes a really big difference. And it's going to really set the trajectory for many careers here. And I was excited because of the young people that I've met here that I already knew. Those would be exactly the people that I would choose to honor. And for those of you who I didn't know, it's been really great getting to meet you. So I want to thank the foundation for that, because I think that is a major service to the science. Now, the thing that I started out working on, which was actually, in part supported by the Dreyfus Award, was using polymers as probes of biology. But I thought I would talk about this area because it's relatively new, because I think one of the things that you can do when you get to the stage where you've established a group and you're off and running is move into new areas. And so that was one of the things that I decided to do. And since I had long been interested in carbohydrates, and the way we actually started using those polymers was to study protein-carbohydrate interactions, I became interested in how carbohydrates are built. And so if you study carbohydrates, or really anything at the interface of chemistry and biology, we can pay homage to Emil Fischer. And I was recently looking at his Nobel lecture, and I found this quote. Actually, it was one of those things where you read it and you think, I so wish that were true. Because so of course he couldn't anticipate how powerful the genetic tools are that allow us to manipulate and study proteins. And he had developed some beautiful chemistry to study carbohydrates, but it turns out biology doesn't really give us the same powerful tools to study carbohydrates, just because we can't easily encode them in the cell. Now, that being said, I will encourage you to talk to Ji Chen, because she's really developing some interesting strategies to build up carbohydrates using biological approaches. But that, because it's difficult to study, suggested that our ability to make molecules would allow us to investigate processes that wouldn't really be accessible to people that only had biochemical, or perhaps more accurately, biological tools to use. So we became interested in carbohydrate polymers. And just to emphasize that this is a problem worth studying, I'll just point out that carbohydrate polymers are the most abundant organic molecules on Earth. They are, of course, found in cellulose. They're found in us. So when this basketball player comes down, his knees have to absorb an enormous amount of force. And the way that you do that is there's hyaluronan, a carbohydrate polymer, in your joints that helps absorb that force. So we have a number of carbohydrate polymers that play important roles. And of course, carbohydrates are really important on microbes as well. And some vaccines are derived from actually the carbohydrate components of pathogens. So these are diverse molecules. They're structurally diverse, and their lengths can be controlled. So, for example, cellulose can be tens of thousands of residues. Hyaluronin, that compound in our joints, consists either of very long polymers in our joints, or there are smaller fragments of hyaluronin that are actually used for signaling. Bacterial polysaccharides can be hundreds of residues or tens of residues. So these really vary in length a lot. And they're not made by template-dependent processes. So I think a very interesting question for this field, and actually for biology in general, are how do you control template-independent polymerization reactions? And then we ask that question in a more local way. We're interested in how mycobacteria assemble an important, an essential carbohydrate polymer. So we focused on mycobacteria, and I really had two aims in this. Frankly, we focused on it because we were very interested in the structure of a key polymer in their cell wall. So here's a schematic version. This is the inside of the cell. This is the out. And what you have is a lipid region, and then this ladder-like structure represents the peptidoglycan. Attached to that is this polymer of galactofuranose residues. Attached to that are arabinofuranose residues, and then these lipids, and then a capsular polysaccharide. So this is actually a different kind of cell wall than typical gram-positive bacteria. And one of the unique features was this galactofuranose polymer. That's really what intrigued us. So we were interested in the role of galactofuranose. And of course, we know that galactose can exist in the pyranose as well as the furanose forms. And in all of our glycolipids and glycoproteins, we use this pyranose form. We never use the furanose. And the fact that this furanose is found in various organisms suggested that the enzymes important for its incorporation would be interesting targets. So where is it found? Well, it's found in a number of bacteria. So this is just standard E. coli. As I mentioned, it's found in mycobacteria. It's also found in some protozoan parasites. So it's not restricted to bacteria. It's found in eukaryotes like fungi and these trypanosomes. And more recently, a gene that's critical for Galactofuranose Incorporation was found in this multicellular eukaryotic organism, C. elegans. So we know it's found in really diverse organisms, but not found in humans. And it turns out all of these are pathogens for the developing world. So we thought this would be an interesting pathway to study. And so this just, I think that we all know that TB is a major problem. It ranks number 2 behind HIV/AIDS for causing the most number of deaths. And just to emphasize this, the scary aspect of it, there's been an extensively drug-resistant form that was identified in October 2006. This form is especially virulent, and it's not treatable with any of the known antibiotics. So TB are harder to treat than many bacteria, and that is, it's thought because their cell wall is, in part, it's because their cell wall is so strong. And I really like this slide that was made by Becca Splain in my lab. So this rocket engine produces 3 times 10 to the 5th nanometer or newtons per meter squared of force, and you can push on these mycobacteria with an AFM tip, and they will resist more force than this rocket engine generates. So what that tells you is there's some unique features of the cell wall that make it really impermeable. And to us, that made it also a very interesting problem to study. Like, why is this cell wall so strong? And, why did the bacteria go to the trouble of putting galactofuranose in there? So I'm not going to have answers to those questions, but hopefully you'll see that it's an interesting question. So the part of the pathway that we're interested in is shown in the context of this larger schematic. And this is really work from McNeil and Brennan's lab at Colorado. They've outlined many of the players in the pathway. Most of these enzymes have not been well characterized biochemically. But essentially what starts is you have a C55 lipid that gets modified by 2 sequential glycosyl transferases to make this disaccharide. Then there's an enzyme, GLFT1, that installs a couple of galactofuranose residues, and that's followed by a polymerase that makes a polymer. And then this is all occurring on the inside of the cell. There's a transporter that takes this to the outside of the cell, again, not very well characterized. And then there are arabinofuranocil transferases that modify this before the mycolic acids are attached. The only target in this entire pathway that is an antibiotic target is this, these arabinofuranocil transferases are targeted by ethambutol, a known clinically used antimycobacterial agent. So there's a lot of room for study here, and we thought as a prelude to that, let's begin to examine the enzymes involved. So let me show you this at a chemical level. There's an enzyme that first generates an activated form of galactofuranose. This is what's going to be used now by the transferase to add galactofuranose residues. So here's our lipid. R is our lipid-linked disaccharide. And then there's this enzyme GLFT1 that attaches 1 or 2 galactofuranose residues, followed by the enzyme that makes the polymer of about 20 to 40 residues. So what I want to do first is tell you a little bit about the enzyme that we've most studied, and that is the enzyme that generates the activated UDP galactofuranose. So this enzyme is known to be essential. If you knock it out, the bacteria are no longer viable. And we designed a synthetic route to this species because it's very difficult to generate biochemically, in that the equilibrium lies on the side of the pyranose form, as you would anticipate. So we want to take 100% of this and get 94% of that, so we chemically synthesize this species. Now, the enzyme was really intriguing to us because it's a flavoenzyme. And flavin is vitamin B2, the second B vitamin isolated. Flavoenzymes have been known for over 70 years and very well studied. And flavin is just a remarkable cofactor. It can exist in a variety of oxidation states shown here. It also has various protonation states. So it's extremely versatile in terms of the chemistry that it can do. Now, what was interesting in this system is that Jim Naismith's lab had shown that it was the reduced form of the enzyme that was active. And so let me just show you what reduced flavins and oxidized flavins typically do. We anticipated the oxidized form of the enzyme might be important initially because oxidized flavins can oxidize things like hydroxyl groups or amines. Reduced flavins typically reduce things like disulfides or molecular oxygen. And when we looked at the substrates, it wasn't really clear how reduction would facilitate this isomerization. So this was just an intriguing chemical problem that drew us in. So there are some flavoenzymes that don't use the flavin in catalysis. They use it mainly as a structural role, but our enzyme didn't fall into that class. And indeed, we were able to solve a structure of the enzyme bound to UDP glucose in collaboration with Katie Forrest's lab. So we know that the flavin is in the active site and it's actually near the substrate, that the enzyme must be reduced to be active. And then something that I didn't show you, but from John Blanchard's lab, we also knew that UDP is a transient leaving group in the process. So these three people set out to study this mechanism. And Erin Carlson is a very talented person who just recently started her independent career at Indiana. Todd is currently a postdoc at NIH, and Michelle is vice president of her company. So it was a great group of students who started this. And so to come back to the mechanism, what we proposed is that perhaps the reduced flavin was acting as a nucleophile to capture the sugar. That would allow the UDP to serve as a leaving group while providing the enzyme with a way of holding on to the sugar, and then the enzyme could catalyze this isomerization via the iminium. So that seemed a reasonable possibility to us chemically. The problem was that these two steps had not been really reported in terms of flavoenzyme catalysis. So Tom Bruce's lab had reported that this sort of intermediate could form chemically, but it had never been observed in catalysis. So we decided to see if we could chemoselectively trap this intermediate. And this was a difficult task because this is itself prone to further oxidation. But again, I was fortunate to have talented students, and Michelle and Todd were able to isolate a new product. So I should explain that the flavin is bound non-covalently. So we can add substrate, reducing agent, precipitate the protein, and then look to see what's in the supernatant. And when we did that, we saw that there was this new product that was different than the flavin. When we took a mass spec of that new product, it had the molecular weight to match the reduced cofactor adduct. And we have additional data that support that this is the intermediate. Needless to say, this experiment is basically an endpoint analysis. And there were many in the community that were, or perhaps remain, skeptical as to this being the mechanism. So we wanted to see if we could provide some structural data in support of this mechanism. So Todd was able to crystallize the enzyme, and then he basically soaked in a substrate. And when we got that structure, initially it was rather actually depressing in that we could see this is the UDP here, and then you can see that this is the nucleophilic position that we were anticipating for the flavin, and it's not at all poised for catalysis. But this enzyme was the active enzyme. And so Katie Forrest, our crystallography collaborator, and I suggested that Todd soak in reducing agent. And when he did, the position of the sugar moved. And now you can see here's the sugar. It's really nicely poised for that chemistry. And so this just shows a close-up view of that relevant portion of the structure. And what was also really nice is David Sanders, Jim Naismith's former student, solved the crystal structure of a homologous enzyme from a different species, and they also found this sort of close arrangement. In fact, their structure was even closer between this N and the C1 of the sugar. So we think that this is reasonable to suggest that in addition to this position of reduced flavin acting as a nucleophile, the N5 position also can act as a nucleophile. And there are indeed some other flavoenzymes for which this kind of nucleophilic nitrogen may actually play a role in catalysis. So that was really what drew us into the program. And so let me just tell you with a really brief scenario that we also have looked at trying to inhibit this enzyme as a target for antimycobacterial agents. We wanted to see if this pathway would actually be useful. And so we did a high-throughput screen, and then off of the hits from the screen, Emily Dijkhuizen in the lab designed new compounds, and we tested these in just an assay where we have on this white disk on a lawn of mycobacteria, we tested whether or not we got a zone of clearance. So that's indicative that this compound, and here's our DMSO control, so ED103 here is actually preventing mycobacteria growth. And we could measure an MIC value for that. It's about 150 micromolar, so it's not incredibly potent, but it's around the same as known antimycobacterial agents. I don't think that these are the compounds to test, but we think that this helped validate this as a reasonable target. And we wanted to then see whether or not these compounds would actually alter the cell surface. And so these are AFM images just of Mycobacterium smegmatis, untreated, and then you can see the defects in the cell, in the outer part of the cell that occur upon treatment with a compound. So we think it's actually blocking the portion, it's creating ridges in the mycobacterial cell wall, which is similar to what you see with ethambutol, another inhibitor of this portion of the cell wall biosynthesis. So that then led us to come back to the question that I started out. How does nature control the length of polymers? And so we wanted to use this galactofuranose transferase as a model for beginning to study that. So I'm going to tell you a little bit about this enzyme. This is a fascinating enzyme to me because it does something that most glycosyl transferases don't. It forms two kinds of linkages, and it seems to do so with sequence specificity in that GLFT2 was reported to take this disaccharide that had been, to which a few GalF residues had been appended, and make a polymer. And you see that here is the 1 position of the sugar. This monomer goes from 1 to 6, and then linkage at the 6, now back to the 1 position, and then here it's the 5 position that forms the next linkage. So this enzyme was reported to make 2 different kinds of linkages in an alternating manner. And again, make a polymer of specific length. So we were really interested in, first of all, whether or not a single enzyme could do that. And so the people who worked on this were Becca Splain, who synthesized the molecules, and John May and Todd Gruber. So Todd, I already told you about his work. This is John, who did the work I'm about to tell you. And just as an aside, I'll just mention this because I love this picture. John is actually putting a necklace on Todd. This is at Todd's defense. These guys started in my lab the same year. They picked the same project. They graduated within 1 month of each other. And when they graduated, my lab bought them each, those necklaces that each have half of the broken heart. So we just referred to them as John and Todd, John and Todd, John and Todd. So what John did was he had cloned GLFT2 out, and we were waiting for then substrates to use. Now, we are working very hard to use enzymes to build up this natural substrate. We've actually looked at both synthetic methods and enzymatic methods to make this. But for the interim, we wanted a compound that we could test, so we made this simple disaccharide precursor. And it had been shown by Bezra that compound was active in, a compound like it was active in microsomal preparations. So we thought that perhaps this would work. We synthesized this, and I'm going to give some foreshadowing here. Using cross-metathesis, we'll probably hear a lot of cool chemistry for metathesis soon. But we synthesized this so that we could, at a late stage, put in different R groups. That's the basis for the design, because we really didn't know what was going to be required at this end. So once we had these compounds, we took and mixed them together, and then we monitored polymerization using multi-mass spectrometry. And what you can see here, each of these numbers refers to the addition of another residue. So we get a polymer. This was an overnight incubation, so not a kinetic study. We let this go till it was complete. And we saw that we got polymeric products. And you see that these alternate. And without showing you the data, we've isolated shorter fragments and shown that these do consist of the alternating 1,5, 1,6 linkages. And you can see that the alternation in the mass spec weights also suggests we're making 1,5 and 1,6 linkages. So that was exciting because it suggested that this enzyme on its own can make a polymer and it can make these alternating linkages. Now, we wanted to ask, does it have an intrinsic ability to control length, or did we just see a random mixture from a single experiment? And so we wanted to ask, does the number of carbohydrate residues itself influence how long of a polymer we make? And so we took and made shorter fragments of various lengths and then re-exposed these to the enzyme. And so the data look like this. If we use n equals 2, we get this pattern, n equals 4, this pattern, n equals longer, the same pattern. And so that suggested that the enzyme does have an intrinsic ability to control the length, at least with this substrate. So we next wanted to ask, well, how does this enzyme work? Is it distributive or processive? Because that would give us insight into how length is controlled. So for a distributive enzyme, it's going to take and bind substrate, do a round of catalysis, and then the substrate's going to fall off, rebind. So there, you can control length by controlling how much of the substrate is available. For a processive enzyme, it's going to take and bind substrate and just continue to add residues. And so those two possibilities would lead to very different mechanistic outcomes. And so one way to look at this is to do single-hit kinetics. And so we used a large excess of substrate such that enzyme would only see substrate once, presumably. It couldn't rebind a partially elongated substrate because that would be there in such minuscule quantities. So we used 1,000-fold excess of substrate, and we saw long products right away. But we did see, you can see that we do see a polydisperse mixture, and so we wanted to look at this more precisely. So Matt Levengood, a postdoc from Wilfred Vanderdonk's lab, and an awesome postdoc, came up with this idea. He said, well, how about if we start the enzyme with a substrate for a specific time, short time, and then add a competitor substrate. And if we can tell the difference between what we started with and the competitor, we can then ask, can this guy compete with this once it started polymerizing? If it can't, it would be processive, and if it can, it would be distributive. So the substrates we made were just labeled deutero and proteo. So we thought this won't affect the chemistry where the reaction's going on. And we ran the reaction for 30 seconds first with this light substrate, and then doped in the heavy substrate and ran for 45 seconds. So here's what we saw. If you look at short products like the 7-mer, there's not really much difference between light and heavy. But if you look at longer products like this 13-mer, we can't really see the heavy in there. And if you run this reaction non-competitively, that is, run the first reaction for 30 seconds longer than the second, and just mix the two samples and look at them, we don't see any big difference. So that suggests that this reaction indeed is processive. And we can use this assay to start to get information about the processivity parameters. How processive is it after the various steps? And so these are some of the data that we got. You can see that there is a lag at the beginning where it's likely that the substrate falls off, and then it becomes more processive as the reaction goes. This isn't a highly processive enzyme reaction, but it is reasonable. It's similar to some of your lousier DNA polymerases. the other thing that we saw is GLFT2 exhibits a lag phase. And this is really characteristic of processive enzymes. So RNA polymerase has a lag phase, polyadenylation has a lag phase, ubiquitination has a lag phase. And the origin of that lag phase is presumably that these enzymes have subsites. And so you need to first have a substrate that's long enough to fill all those subsites, and then you get elongation. And so we can eliminate that lag phase by making short oligomers. And this model predicts the enzyme has a single active site. And since I don't really want to, how am I doing for time? Bad? Okay, I'm skipping these. Okay, the enzyme has one active site. Okay, I'm going. So now we'll ask, how is the length controlled? One of the interesting things that we saw is that this R group really made a big difference. So the enzyme has a single active site, but when we changed this R group, so we made a shorter R group, we saw that in that case, only 4 GalF residues are added. So the enzyme is processive, but it depends on the nature of this lipid. And the rate doesn't differ that much in processing these different substrates, but the K1/2, which is similar to a Km, is actually different for these two. So the lipid is influencing the Km. And this is different than the model that we had here, where you just have the lipid out here and all you care about is occupying all the subsites. This kind of model doesn't allow you to predict how you would actually control length in any way, because what's to stop the chain from continuing to elongate? So we thought, well, since the lipid makes a difference, one way in which this enzyme could control length is to have subsite binding, so a lipid binding site distal from the site where catalysis is taking place. And if that occurs, then when the chain can no longer accommodate this dual binding, it will fall off. And so that would be a way that one could control length. And so to test for that, we thought, let's just make a lipid that's longer that should more effectively occupy the site, increasing effectively the Km, and so we could maybe make a longer polymer. So we started out with a lipid of 9 groups. I already showed, of 9 methylene groups, I showed you those data. We made another substrate that now had a longer lipid, and you can see that we can make longer polymers. The kind of neat thing about this idea is if it's how nature works in this system, you could imagine then toggling the length of oligosaccharides by toggling the affinities of these different components for the enzyme. And I think that's an attractive idea that would allow systems to evolve with different sorts of control mechanisms. So just to conclude, what I've told you about is first this mechanism where flavin plays this different role in catalysis in the case of UGM, and then the design of inhibitors that block elongation, and then lastly this tethering mechanism. So I'll just end by saying that there are a lot of other polymers that nature makes. And one thing that was fun is I gave this talk at Colorado, and Tom Check said that he has come up with a similar mechanism for telomerase, where there's, again, a distal binding site that controls access to, or that facilitates access of a chain of strand to the enzyme active site. So perhaps this idea may be one that's actually broadly used in a variety of systems. So I've tried to thank the people who did the work throughout the talk. I want to thank them, and I want to thank Mark and everyone for the opportunity to come here and tell you about this work, and I'd be happy to take any questions. David has a question.
Thank you, Laura. David Hansen from the Joint Science Department. At this point in time, here in 2010, what do you see the relationship between academic labs, big pharma, biotech perhaps, in terms of the development of small molecule drugs and bringing them into the clinic?
Wow, that's a complicated question. So what I will say about this is that from the academic point of view, I really am interested in the compounds that we found as probes and not so much as drugs. I'm happy for pharma to take them up as drugs. And again, there's not a lot of research going into mycobacterial infections because they largely afflict the developing world. But I think if you have something, people are happy to pick up the banner and go with it. And if you can validate targets or give a promising lead, so I'm answering this in a very local way because I can't really answer in a global way. But with regard to probes, we are collaborating with the Broad to try to come up with probes that would block these steps in the pathway. And the reason I'm interested in that is because I'm basically interested in how defects in the cell wall might influence host pathogen interactions, immune responses. And so I think that there's a lot of great science that we can do in academia that will then be useful to people in pharma or biotech. But I'm not personally that interested in developing a drug myself.
On its own. So a simple question after the last one. So can we think about this control maybe as a surfactant kind of thing? So you've got a hydrophobic and you're putting on a hydrophilic end, and so it's a balancing between those effects. And so basically when you get the hydrophilic big enough, it just dissolves and goes away. Is that a...
So I think that's an interesting idea. So I should say we've looked at all these compounds below their critical micelle concentration because one of the things we were worried about is we're just sort of solubilizing the enzyme that the enzyme needs to interact with a membrane. And so I'm not sure I'm answering your question, but we know that critical micelle concentration is not the key parameter. In other words, we have compounds that form micelles more readily and are polymerized less effectively or make shorter polymers. That it really had to do with a lipid length. So the question of whether it's a specific recognition or sort of a general hydrophobic, hydrophilic, I don't think I can answer that definitively. We're really trying to do experiments to address that. But one of the things that really, I recently went to a talk by all these people who work on, I went to a bunch of talks by people who work on G proteins, and those have these polyprenol modifications. And then there are proteins that then ferry these around the cell. And so what has to happen after we make these polymers is that it has to go to the transporter and make it to the outside. So it's possible that pulling that lipid out of the membrane a little bit then might facilitate that handoff. This is total speculation, but since you're not, don't put this part on the web or anything.
So, but many of these proteins have protein-protein interactions. So the question is, are you missing the transporter, the WZM that you had? And the other question is, what the heck is going on with the glucose structure that you got? You just have a big loop hanging out in solution. What do you expect?
Yeah, so
with that? So
those are two. Those are two. I just draw it that way because I don't know what the structure is, but I'm pretty convinced that there's a structure to that alternating 1,5,1,6 compound. We've done some modeling, and it looks like, if you made all 1,5, it looks like the molecule becomes very quickly too cramped to make actually additional linkages. If you make all 1,6, it looks really extended and flexible. And so maybe there's really a specific structure that's needed. So we are working really hard to get structural data on that. As for the WZM, the control mechanism, there are no homologs in mycobacteria. So we've really looked hard to see. So this is just for, so Joanne and I don't sound like we're talking in code. There were some really nice results published from George Wang's group that showed in E. coli there actually is a molecule of protein that controls length of and a similar type of carbohydrate polymer synthesis that tells that, you're done, that's long enough. So we've really looked hard for anything homologous, and there isn't in this system.